Okay, hello to all of you. Let's discuss chapter number 6. So, chapter number 6 is basically about the cell, and we are going to take the tour of the cell and we are going to discuss some of the important organelles. So, let me go into the slideshow mode. Okay, so basically, as you already know, the cell is the basic unit of life, and all the organisms, they are made of cells, and the cells, they come from the pre-existing cells. And we have basically two kinds of cells, prokaryotic cells and the eukaryotic cells. So we have, I think, discussed in first chapter also what are prokaryotic cells and what are eukaryotic cells. So the word karyota means nucleus, and the word U means true. So eukaryotic cells are those cells which have a true nucleus, and prokaryotic cells, pro means before nucleus, or basically prokaryotic word basically comes from, the meaning is that they don't have true nucleus, and these are the cells which were there before the nucleus came into being. So so prokaryotic cells do not have a true nucleus. Eukaryotic cells, they have a true nucleus. So, but do prokaryotic cells have genetic material? Yes, prokaryotic cells, they have genetic material, because that's a basic requirement of life. Because all the organisms, they reproduce, they pass the characteristics to the next generation, and that can happen only through the genetic material. So prokaryotic organisms include bacteria and archaea. So these are the two. So we have talked about the tree of life. There we talked about there are three domains. There's a we have discussed all these things in the first chapter. We talked about bacteria, archaea, and the eukaryota. And bacteria and archaea, these are prokaryotic organisms, or prokaryotic organisms or prokaryotic cells. And the eukaryota basically have the eukaryotic cells. And eukaryotic cells include animals, plants, fungus, and protists. Okay. So whether it's a prokaryotic cell or it's a eukaryotic cell, all the cells, whether prokaryotic or eukaryotic cells, they all have plasma membrane. We will discuss about plasma membrane in detail in next chapter, but plasma membrane is a boundary which separates the cells from its environment. So all cells, they have plasma membrane. They all have semi-fluid material in the cell, that's called cytosol. Then all have chromosomes which carry the genes, and they all have ribosomes which make the proteins. So these are the basic features of all cells. Now prokaryotic cells, they are, now let's talk about the characteristics of prokaryotic cells in general. So prokaryotic cells, they don't have any nucleus, and the DNA is present in the cytoplasm, and that region where the DNA is there, it's called nucleoid. And they don't have membrane bound organelles, and the cytoplasm is bound by the plasma membrane. So the most important thing to know about prokaryotic cells is they do not have nucleus, but they do have genetic material, and the genetic material is present in the cytoplasm, and that region where the genetic material is there, it's called nucleoid region. So here is an example of a bacterial cell. So you can see this is the bacterial cell. This is the electron micrograph of a bacterial cell. This is a picture taken by the electron microscope. So the bacterial cells, they have a cell wall, okay, and they have a cell wall. So cell wall, and most of the bacterial cells, they have a cell wall. There are only few cells, few exceptions. Mycoplasmas are the ones which don't have the cell wall. So they have a cell wall, then they have a plasma membrane, and then inside you can see this is all cytoplasm. And this is the genetic material, this is the chromosome, this is a bacterial chromosome. And this region where this bacterial chromosome is there in the cytoplasm, this is called a nucleoid region. And some of the bacteria, they also have capsule around them. And this capsule which is there in the bacteria, it basically provides protection to the bacteria. So when the bacteria infect the host, because some of the all bacteria do not have capsule, some of the bacteria have capsule. So this capsule basically protects the bacteria from the attack by the immune system of the host. So the bacteria which have capsule around them, they are very virulent and pathogenic bacteria. And the bacteria, they have flagellum which helps them in the movement. And then around the surface of the bacteria, you can see these are short appendages, and they're called fimbriae. And fimbriae basically helps in Fimbriae basically helps in the attachment of bacteria to the cells of the host. So this is the basic structure of the bacterial cell, which is a prokaryotic cell. Now, talking about eukaryotic cells, the most important thing to know about eukaryotic cells is eukaryotic cells, they have a true nucleus, and in the nucleus, which is the largest organelle in the eukaryotic cells, in this nucleus is the DNA is there, the genetic material is there. So they have a special organelle in which the genetic material is there. And other than nucleus, these eukaryotic cells, they also have other membrane enclosed organelles which are not there in the prokaryotic cells. So this is the main difference between eukaryotic and the prokaryotic cells, and eukaryotic cells are generally much larger than the prokaryotic cells. Now let's talk about some of the important things which are there. First of all, plasma membrane. So we are going to first of all talk about some of the common things which are there in both, and then we are going to talk about some of the special organelles which are there in the eukaryotic cells. So plasma membrane is there in both the prokaryotic cells and the eukaryotic cells, and plasma membrane is basically the boundary which separates the cell from its environment, and through the plasma membrane there is an exchange of gases taking place, there is an exchange of various solutes taking place, various ions are crossing. So this is the structure of plasma membrane. We will talk about in chapter number 7. So plasma membrane is made of phospholipid bilayer. So we discussed in chapter number 5 about the lipids, and one of the category of lipids is phospholipids. So we also talked about there that phospholipids, they have an amphipathic nature, which means they have a dual nature. They have a polar head as well as they have non-polar tails. So they have a polar side as well as they have a non-polar side. So we also discussed there that if we take the phospholipids and if we put them into a glass of water, how they're going to arrange themselves. So we talked about that the polar head of the phospholipids, it is hydrophilic in nature, which means it is water loving, and the non-polar tails, they are hydrophobic, they are afraid of water. So if we take a glass of water and we put these phospholipids in the glass of water, these phospholipids, the non-polar tails, they actually face towards each other, and the polar heads they stay outside because they can face water because they are hydrophilic. So this is the way the phospholipid bilayer is made. And also we discussed in the cell, inside the cell, if this is the inside of the cell, there is all cytoplasm. Cytoplasm is a jelly kind of a material and which has a lot of water in it. So inside the cell is water, outside the cell is also water. So there is a water on both sides. So these phospholipids, they again arrange in a bilayer. They form a bilayer like this. So the plasma membrane, which is a boundary which separates the cell from its environment, and inside the cell there is water, outside the cell there is water. So these phospholipids, they arrange like this, they form a bilayer, and in between this bilayer are embedded some proteins. Okay, and these proteins, they do many functions. They act as enzymes, they are transport proteins, they are receptors. We'll talk about all these things. So this is the basic structure of the plasma membrane. So this is the hydrophilic region, this is the all hydrophobic region. Okay, important thing to know here is, as we're talking about the membrane, here I would like to ask a question. If I would have been teaching in the class, I would ask a question here, like if you see a small animal like a cat, or if you see a big animal like an elephant. Okay, so elephant is very big, cat is a small animal. So both are animals. So I would like to ask my students a question here. What do you think? If elephant is a big animal, do you think the cells in the elephant are big as compared to the cells in cat, which is a small animal? And I would let you guys think about it. So basically the answer is no. In the big animal like elephant, they do not have big cells, but they have more number of cells. Okay, small animal like cat, it's not that their cells are small. Okay, so there is a limit and beyond that limit the cell size cannot increase. Okay, so basically it's not that in the big animals they have big cells, but they have more number of cells. The reason is there is a limit, and beyond that limit the size of the cell cannot increase. Okay, now why the size of the cell cannot increase? The reason is, as we are talking about the plasma membrane, through the plasma membrane there is an exchange of material going on. Things are moving in, things are moving out, gases are getting exchanged through this plasma membrane. So there is a ratio called surface area to volume ratio. There is a surface area to volume ratio that needs to be high in the cells, because if that ratio is not high in the cells, then this exchange will stop. Okay, this exchange will stop, and if this exchange of the material which is going across the plasma membrane, if that stops, then the cell will die. So it's very important to maintain the surface area to volume ratio high. So surface area to volume ratio, if it is high, what does that mean? It means that there is a more surface area, of the cell which is there, through which the exchange can take place. But if there is less surface area through which the exchange can take place, then the cells are not exchanging these material and the cell is going to die. So that is why there is a limit beyond which the cell cannot increase its size. The reason is, when the cell increases its size, so if you talk about surface area to volume, so it's all written here. This is the formula for calculating the surface area. So basically it is height into width, and volume is always height into width into length. So this is the square value, this is the cube value. So if you take the ratio, surface area to volume ratio, so volume is cube ratio and surface area is square ratio. So the volume is a denominator. So as the size of the cells increases, increases, the volume also increases. If the volume increases, denominator increases, this ratio decreases, which means that the effective surface area, which is required for the exchange of the material, that decreases, and if that decreases, the cell dies. So that is why the big animals do not have big cells, they have more number of cells. So here is example, you can see this is a small cell, okay, and this is a big cell. So if you take this cell, its dimensions are 1, and if you take so basically means its height, width and all that is 1, unit 1, and this is a big cell, its dimensions are 5. And if you use these formulas and you calculate, the surface area is 6, volume is 1, and if you calculate the surface area to volume ratio, it is 6. Then if you take this big square, then its surface area is 150 and its volume is 125. So the surface area to volume ratio, this ratio becomes 1.2, which means it decreases. So it means the effective surface area decreases. But if we have this kind of arrangement, like if this is a big animal, if this is a situation, if the big animal has one big cell, but on the other hand, the way the actual arrangement is, the big animals, they have more number of cells. So these are the number of cells, these are the different cells, and if you calculate the surface area and volume ratio, in this arrangement it comes to 6, which is equivalent to this. So in this arrangement they have more effective surface area. Okay, so now we are going to talk about the eukaryotic cells. So in the eukaryotic cells, we will be talking about basically the two important kind of eukaryotic cells, the plant cells and the animal cells. So overall, I'm going to show you in short. This is the animal cell. So you can see animal cell doesn't have a cell wall. So this is a very important difference between animal and the plant cells. Animal cells do not have the cell wall, and plant cells, they do have the cell wall. Okay, so they do have the cell wall. So these are the different organelles in the eukaryotic cells. So they have a nucleus. Nucleus is considered the brain of the cell, because all the information, genetic material is here, genetic information is here, and it's covered by the plasma membrane, it's enclosed by the nuclear membrane, and this nuclear membrane extends into the cytoplasm. It extends into cytoplasm, and it's called endoplasmic reticulum, which is a network of the membranes. Then this is Golgi apparatus, and this is mitochondria. Then so these are different organelles. We are going to talk about all these in detail very soon. Okay, so you can see all these are membrane enclosed organelles. Okay, and this is the plant cells. The main difference between animal and plant cells is plant cells have a cell wall. They have a central vacuole. In this central vacuole, they can store a lot of water, various organic things, and then these are the chloroplasts. And in the chloroplast, the photosynthesis takes place. So these are the three main differences between the animal cells and the plant cells. Okay, so now let's talk about all these organelles. So first of all, the nucleus. So nucleus is the largest organelle in the eukaryotic cells. It's a brain of the cell, and your DNA is there. Okay, so let's go and talk about here. So this is the nucleus. So in the nucleus there is your DNA, and important thing to know about DNA is it is wrapped over the proteins, and those proteins are called histone proteins. So these are histone proteins. So this is very, very important thing to know here, and we will talk about this again when we talk about, I think, chapter on replication, DNA replication. So DNA, so very important thing to know here is DNA. Basically, DNA is a very, very long molecule inside the nucleus, and because DNA is carrying so many genes, and which is controlling us. So the cell is very small, and nucleus is even smaller. So how does this all long molecule of DNA gets fit into the small nucleus? So this DNA is wrapped over these histone proteins. Okay, so these are histone proteins. So the DNA is wrapped over them, and it gets all condensed. Okay, so it's all gets looped, and it becomes very tight. We call it it gets condensed, and it forms chromatin. Okay, so it forms chromatin. So the DNA inside the nucleus is wrapped over the histone proteins. So DNA is wrapped over the histone proteins, and it's very, very tightly coiled, and it is called chromatin. And this region in the nucleus, is called nucleolus. Nucleolus is a region where rRNA is made. Okay, so we will be talking about ribosomes, which are basically the complex structures which are made of rRNA. What is rRNA? rRNA is RNA, and proteins. So this rRNA is actually made here. Okay, ribosomes are there in the cytoplasm. They are in the two forms, the free form or the bound form. We're going to discuss shortly. That rRNA is made here. Okay, and this nucleus is enclosed by the nuclear envelope, and in this nuclear envelope there are some pores. They're called nuclear pores, and the nuclear envelope is bilayered. It has the outer membrane, it has the inner membrane, and in between there are pores. And through these pores, transfer of things takes place. mRNA is also made in the nucleus, and it goes to the cytoplasm through these pores. So you can see here. So this is the basic structure of the nucleus. Okay, so this is nucleolus. Chromatin is here. This is the nuclear envelope. Nuclear envelope has two layers. It has the outer membrane, inner membrane, and in between there are nuclear pores. And I would like to tell you one more important thing is about chromatin. So chromatin is very condensed genetic material. So it's a DNA wrapped over proteins. So if you see a cell when the cell is not dividing, so if you see the cell, if you view the cell under the microscope, and if the cell is not dividing, then you will see a dense mass inside the cell, and that is called chromatin. But when the cell starts dividing, okay, when the cell starts dividing, then this chromatin gets more condensed. Okay, this chromatin gets more condensed, and it takes the shape of chromosomes. So every species has a specific number of chromosomes. Like humans, we have 46 chromosomes, and these chromosomes, they have a specific shape. Okay, so if the cell is not dividing, then you will see a dense mass. You will not see that specific shape of the chromosomes. You will see the specific shape of chromosomes only when the cell is dividing. So all these things we will talk about in the lab part. Okay. So also this nuclear envelope is basically having some proteins in it, and we call it a nuclear lamina, which gives a shape to the nucleus. So this is what I explained. So the nuclear size of the envelope is lined by nuclear lamina. The nuclear size of the envelope is lined by nuclear lamina, which is composed of proteins and maintains the shape of the nucleus. So I have already talked about in the nucleus, DNA is organized into discrete units called chromosomes. So chromosomes will be only visible when this chromatin will get condensed during cell division. Okay. So this is all I have discussed about all these things. Okay, the next topic we need to discuss a bit about is ribosomes. Now, what are ribosomes? So they are complexes made of ribosomal RNA and protein, and we just discussed this ribosomal RNA is made here in the nucleolus, which is a region in the nucleus. And there are so basic function of ribosomes is the proteins are made on the ribosome. The most important function of ribosomes is proteins are made on the ribosomes, and ribosomes are also called protein factories. And ribosomes can be in the cytoplasm, so they can be free ribosomes, or they can be these ribosomes can be present on the endoplasmic reticulum. Then they're called the bound ribosomes. So these ribosomes can be present sitting free in the cytoplasm, or they can be embedded on the endoplasmic reticulum, and then they're called the bound ribosomes. So this is the basic structure. So you can see this is electron micrograph. So you can see these are the free ribosomes which are present in the cytosol, and there are some ribosomes which are present, which are embedded on the endoplasmic reticulum. So they are the bound ribosomes. And this is the structure. This is the structure of the ribosome. So the ribosome has a large subunit and it has a small subunit. Okay. So now we are going to talk about endomembrane system. So what does the endomembrane system means? So basically our cell is nothing but a membrane. Okay. So our cell is surrounded by a plasma membrane which separates the cell from its environment. Okay. So the outermost boundary of the cell is made of plasma membrane, and this plasma membrane is made of phospholipids. So plasma membrane, the membrane which surrounds the cell, is part of endomembrane system. And then the nucleus, and then the nucleus is surrounded by nuclear envelope. Okay, and then this nuclear envelope actually, this nuclear envelope. So this is the nuclear envelope. You can see this is the nuclear envelope. It's the nuclear envelope, and this nuclear envelope actually extends. It actually extends into the cytoplasm, and this nuclear envelope actually extends into the cytoplasm, and it forms the network of membranes, and this is called endoplasmic reticulum. So endoplasmic reticulum means, reticulum word means network. Endoplasmic means the network of membranes inside the cell. So endoplasmic reticulum is nothing but the extension of the nuclear membrane in the cells. Okay, so this is what is called endoplasmic reticulum. So now endoplasmic reticulum can be of two kinds. It can be smooth as well as rough. We are going to talk about that. And then, so endoplasmic reticulum is also part of endomembrane system. Then we have another organelle called Golgi apparatus. Then we have lysosomes, vacuoles. So all these organelles, they are nothing but they are membranes. Okay, and they're performing different functions in the cells. Okay, and they form the endomembrane system. And the whole endomembrane system is connected, is all connected in the cell, and it is connected in the cell through vesicles. Now what are vesicles? They are small membrane sacs. Okay, so I will tell you what exactly vesicles are and how this endomembrane system is connected very shortly. Let's first of all, let's go here actually. Here. From here, I would like to explain you that this is the plasma membrane. We just talked about the endomembrane system. This is the plasma membrane. Okay. Oops. This is the nucleus. This is the nuclear envelope. This is endoplasmic reticulum. Okay. This is nucleus. This is the nuclear envelope, and this is the endoplasmic reticulum. This is the Golgi apparatus. Okay. And then, okay, we are not shown lysosomes here. Okay, so these are the important components of the endomembrane system. So I would like to show you how this system is connected. So suppose there is some protein made here on the endoplasmic reticulum, because endoplasmic reticulum is embedded by the ribosome. So the function of ribosomes is to make proteins. So if a protein is made here, or the function of smooth ER is to make lipids, or a lipid is made here, now they are not fully made in the ER. They need to go to Golgi apparatus to be modified more. So how does the protein from here moves from ER to Golgi apparatus? They move from Golgi apparatus. They move from ER to Golgi apparatus with the help of these vesicles. Now, what are vesicles? This is a vesicle. Now, what is a vesicle? It is a small membranous sac kind of a structure. And how it is formed? So remember that this is all endomembrane system. Plasma membrane is a membrane. Then nuclear membrane or nuclear envelope is a membrane. This is endoplasmic reticulum. This is also membrane. This is Golgi apparatus. This is also membrane. Okay. So when something has to move from ER to Golgi, this part is going to this part of this they're called cisternes of the ER. It's going to bud off from here, and when this part of the membrane buds off, it forms a sac kind of structure like this. This is called a vesicle. So this vesicle will move, and it will go and it will join the Golgi apparatus. Why it can join the Golgi apparatus? Because Golgi apparatus is also a membrane. Because of the nature is same. And what are these membranes made of? They are basically made of phospholipids, and some proteins are embedded in them. Because they're all membranes, they're made of basic same structures. So these membranes can bud off, form vesicles, and these vesicles can go and fuse with the membrane here. And this is the way how delivery of things takes place inside the cell. So all the transport of lipids and the proteins inside the cell is taking place through the vesicles. Okay. Let's talk about endoplasmic reticulum. So endoplasmic reticulum is nothing but an extension of the nuclear envelope. And there are two kinds of endoplasmic reticulum, smooth and rough. Smooth ER lacks ribosomes, and rough ER has ribosomes embedded in them. Because the ribosomes are embedded on them, their texture is rough. That is why they're called rough ER. Okay, so basically you can see this is nuclear envelope, and this is the network of membranes which is extending from the nuclear envelope. So the part of the ER which doesn't have ribosomes, the part of the ER which doesn't have ribosomes, it's called smooth ER. And the part of the ER and the part of the ER which has ribosomes on it, the part of the ER which has ribosomes on it, it is called rough ER. It's called rough ER. And these folds of the ER, they're called cisternae, and the space which is there in the cisternae is called ER lumen, and on the rough ER there are ribosomes embedded on it. So the main function of rough ER is to make proteins, and the function of smooth ER is to make lipids. It also metabolizes carbohydrates, detoxifies drugs and poisons, and also stores calcium ions. Okay, so this is the main function of the ER, rough ER and smooth ER. Next is Golgi apparatus. Now, the function of Golgi apparatus. So usually I give an analogy here. Golgi apparatus is just like a post office. Now, what is a post office? So post office basically gets the letters from different regions, right? And on the basis of the zip codes on the letters, the post office sorts these letters, and then they send them to a destination. So similar is the function of Golgi apparatus. So Golgi apparatus basically gets the proteins and lipids which are made in the ER, endoplasmic reticulum, and then it, like if it is a protein, this we have talked about what the folding of protein is, how the chain folds, we have discussed in chapter number 5. So more folding takes place in Golgi apparatus, and also these proteins and the lipids, they can get more modified in the Golgi apparatus. Modification, one of the modification is like glycosylation, the addition of sugar chains to these proteins and the addition of sugar chains to the lipids. So all these things takes place inside the Golgi apparatus. Okay, and so Golgi apparatus modifies products of ER. It manufactures certain macromolecules also, sorts and packages materials into the transport vesicles. So the function of Golgi apparatus is to sort these proteins and lipids and packs them into the transport vesicles, and it sends them to their destination. So that is why Golgi apparatus is like a post office. So what is the structure of this Golgi apparatus? The Golgi apparatus basically consists of flattened membranous sacs called cisternae. So I just showed you the folds of Golgi. They are called the cisternae. So these are the cisternae. And the side of the Golgi apparatus, which is facing towards the endoplasmic reticulum, that side is called cis side, cis phase. And the other side, which is facing towards the plasma membrane, that's called the trans phase. Okay. So cis side is the receiving side, and the trans side or the trans phase is the shipping side. Next organelle is lysosomes. So what are lysosomes? We usually call lysosomes as garbage disposal system. So anything which is not required in the cell is taken care by the lysosomes, because these lysosomes are basically kind of membranous sacs, and they are basically formed from they're formed basically from the Golgi apparatus, and they have some hydrolytic enzymes in them, and those hydrolytic enzymes, they help in the digestion of anything which is not required in the cell. So it's like a garbage disposal system. So anything which is not required in the cell, is taken care by the lysosomes. And these lysosomes, these enzymes which are there in the lysosomes, they work best in the acidic environment. So I can show you here, like this is if something foreign is entering into the cell. So you can see, and this process is called phagocytosis. Phagocytosis is anything solid entering into the cell. So you can see if this is some foreign material, some bacteria or something, if it is entering into the cell, the plasma membrane is going to invaginate. And it is going to form a food vacuole like this. Okay, so anything foreign entering into the cell, the membrane is going to invaginate like this. It's going to form a food vacuole. Now this is not required by the cell because something foreign is entering. So the lysosomes from the cells, they're going to join this food vacuole, and because these lysosomes, they have hydrolytic enzymes in them, they're going to chop all the material in the food vacuole. So it will be taken care by the lysosomes. So another thing is this process, the second mechanism I'm going to talk about is called autophagy. Now autophagy is self-eating. So if there is some organelle inside the cell like a mitochondria, it is an old organelle, it's a worn out cell, doesn't need it. Then what is going to happen? The lysosomes are going to fuse, the lysosomes are going to fuse with the organelle. And it's going to release its hydrolytic enzymes. It's going to chop this. So this is the way lysosomes take care of anything which is unwanted in the cells. Then the next organelle is called vacuoles. So there are different kinds of vacuoles. So plant cells, they have a big central vacuole, and these are basically the vesicles which are derived from ER and Golgi, and they perform different kinds of functions. So central vacuoles, as I said, they are present in the plant cells. Then there are food vacuoles, which I just showed you, which are formed like this by the process of phagocytosis. And then there are contractile vacuoles, which are found in some of the freshwater organisms, because they help in actually pumping the excess water from the cell. So these are different kinds of vacuoles. Okay. So some of the organisms, they live in some environment, and because of which a lot of water can enter into their cells. And the function of these vacuoles is to pump that excess water which is entering into the cell, because otherwise they're going to burst. So these are different kinds of vacuoles. So this is the central vacuole in the plant cells. So I have already shown you how the endomembrane system is connected. Okay, so this is basically a protein being formed here in the ER. It's now getting bud off from here. It's forming a vesicle. It's now this vesicle is fusing with the Golgi apparatus, and all the modifications are going to take place inside the Golgi. And then again this vesicle, is going to be this vesicle is going to bud off from here. This vesicle is going to bud off from here, and then it is going to go to its destination. Like for maybe for this protein, its destination is going outside the cells, because this may be a secretory protein. So you can see how this vesicle is fusing with the plasma membrane. So how this vesicle is fusing with the plasma membrane, and how this protein is going outside the cell. Okay, next two organelles I'm going to discuss. They are not part of the endomembrane system, but they are very, very important organelles, and they're performing very, very important functions. And they are also enclosed by two layers of membranes, and those organelles are mitochondria and chloroplast. So mitochondria, they are basically the sites of cell respiration. So mitochondria is an organelle which is responsible for producing the extracting the energy, from the food we have eaten. And so basically mitochondria is also called the powerhouse of the cell. So it is not that there is some magic taking place in the mitochondria, energy is being produced. No, in the mitochondria, the energy is being extracted from the food we have eaten. So energy is in the food, but the energy is extracted in the mitochondria. Then the next organelle is chloroplast. So chloroplasts are basically found in plants and algae, and they are the sites of photosynthesis. So they are found in plants and algae because they are the ones which do the photosynthesis. And another organelle is peroxisomes. They are basically the organelles which are surrounded by a membrane, and they have some enzymes in them. Okay, something to know about mitochondria and chloroplast is there is a theory called endosymbiotic theory. According to this endosymbiotic theory, that this chloroplast is basically a photosynthetic bacteria. Okay, and this photosynthetic bacteria, it started living inside a eukaryotic cell. Okay, according to endosymbiotic theory, this chloroplast is a photosynthetic bacteria. So there is a photosynthetic bacteria called cyanobacteria. So it started living inside the eukaryotic cell. Okay, and they developed a symbiotic relationship, because this eukaryotic cell, it got the ability to do the photosynthesis, and this bacteria got a house to live. So that's the way how this photosynthetic bacteria became part of the eukaryotic cell, and we call it a chloroplast now. And on the other hand, what is a mitochondria? According to this endosymbiotic theory, mitochondria is basically an aerobic bacteria. Mitochondria is an aerobic bacteria, and this aerobic bacteria actually started living inside the eukaryotic cell, and both again benefited from each other. Both benefited from the symbiotic relationship, because the aerobic bacteria got a house to live, and the cell got the ability of this cellular respiration. And there are many evidence to support the endosymbiotic theory, because mitochondria and chloroplasts, they have many similarities with bacteria. First of all, they are enveloped by a double membrane. So that shows that this mitochondria and chloroplasts, they are actually bacteria, because the one layer is the inner membrane is of the bacteria, and the outer membrane is actually the plasma membrane of the host. So that is why they have, because this bacteria, so if this is a bacteria, you can see how it is entering into the cell. This membrane has to invaginate. So this, you will soon see these organelles, they have two membranes. If this is the membrane of the bacteria itself, so when it entered into the host cell, so this membrane, it invaginated. So this invaginated, and it formed a kind of a vacuole. So the outer membrane is actually the plasma membrane, outer membrane is actually the plasma membrane of the host, and the inner membrane is actually the membrane of the bacteria. So this is there are many evidence to support the endosymbiotic theory, and also these you will see that these organelles, mitochondria and chloroplasts, they have free ribosomes in them, and they also have DNA in them. Okay, because they are bacteria, because bacteria has its own chromosome, so these organelles, they also have their own DNA and they also have free ribosomes, so which basically prove that these organelles are actually bacteria. So there are many more. The size is also like of a bacteria. So there are many evidence to prove that, prove this endosymbiotic theory. So now let's talk about the structure of the mitochondria. So basically, as I said, mitochondria has a outer membrane, okay, and it has an inner membrane. So between outer and inner membrane, there's a space called intermembrane space, and the inner membrane is invaginated like this, and these are called cristae. And why this inner membrane is invaginated? This invagination is to increase the surface area. Okay, to increase the surface area. Why to increase the surface area of? Why to increase the surface area of this membrane? Because there are some very important proteins. Because there are some very important proteins embedded in the inner membrane. So that is why it's required to increase the surface area. And this region in the mitochondria is called mitochondrial matrix. And in this mitochondrial, this is the jelly kind of a material here. And in this mitochondrial matrix are embedded some ribosomes and also the DNA. So this is the DNA. Okay. So this is the basic structure of mitochondria. And then let's talk about chloroplasts. So chloroplasts, they are present in algae and the plants, and they are nothing but photosynthetic bacteria. And this is the structure of chloroplasts. So they have an outer membrane, inner membrane, then they have an intermembrane space, and they have these disc kind of a structures which are called thylakoids. And in the membrane of the thylakoid, is the pigment called chlorophyll. And these thylakoids, they are present as a stack, just like a stack of pancakes, and this is called grana. And this here is all jelly kind of a material called stroma, and in the stroma is the DNA and the ribosomes. So this is basically the structure of the chloroplast. And the next organelle is peroxisomes. They are basically bound by a single membrane, and they have some important enzymes in them. And these enzymes can basically they can convert toxic things like hydrogen peroxide into water and oxygen. Okay, so these are the peroxisomes. Okay, the next topic in this chapter is cytoskeleton, and I think I will stop this here, and I will record it in a separate video.